Okay, so today we will finish chapter 16, finish our discussion of the Diels-Alder reaction. Then we will talk about polyenes and their absorption of light. We'll talk a little bit about how vision works. And then we'll start chapter 17 talking about benzene and its structure. And an important announcement, just a reminder that we will have a quiz on Monday. That quiz will cover the material that we've covered in these 3 recorded lectures. Anything from chapter 16, and then if it comes from 17, it will just be from the first 2 sections. So make sure you watch the videos, read the textbook, work the homework problems within the textbook, and you'll be fine, you'll be ready. Think of it as the summer reading quiz that you would have had when you took AP English or AP History in high school. This is just a quiz on your winter reading that you had to do before the semester starts. Okay, so we're talking about rules governing Diels-Alder reactions. And the second rule we taught you was that having an electron withdrawing group attached or conjugated to the dienophile accelerates the Diels-Alder reaction, increases the reaction rate. And we're in the process of using molecular orbital theory to explain how this works. We talked about frontier molecular orbitals, the HOMO and the LUMO, and we drew the molecular orbitals for ethylene on the board. We said that the lowest energy molecular orbital always has 0 nodes, meaning that the orbitals are in phase with each other as you go across the structure of the conjugated p orbitals. The signs of those orbitals do not change. And then as we increase one orbital in energy, we add a node. So with ethylene, where we only have 2 pi molecular orbitals, because we have 2 p atomic orbitals, we end up with 2 pi molecular orbitals. I should have put a pi here, because we're only considering the pi molecular orbitals. So we end up with one with 0 nodes and one with 1 node. And the pi molecular orbital with 0 nodes is the one that is occupied, the lowest energy molecular orbital. Since we only have 2 molecular orbitals of ethylene, the occupied one is the HOMO, the highest occupied molecular orbital. And the unoccupied one is the LUMO, the lowest unoccupied molecular orbital. On the screen here, we have a figure. This is a figure from another textbook, but it's an important figure, and it is available on Learning Suite. And it shows you here the HOMO of ethylene, 0 nodes, and then here we see the LUMO of ethylene, the antibonding pi star molecular orbital with the node in between the 2 carbon atoms. All right, so now we need to look at the molecular orbitals for the diene, and our simplest diene is 1,3-butadiene. So we'll go ahead and draw molecular orbitals for 1,3-butadiene. This molecule has 4 p orbitals, and so we would use those 4 p orbitals to create 4 pi molecular orbitals, and there will be 4 electrons in these 4 pi molecular orbitals. So we'll make a similar kind of a graph with increasing energy on our y-axis here, and we're going to draw our 1,3-butadiene in the S-cis conformation, because that's the conformation that participates in the Diels-Alder reaction. So our lowest energy pi molecular orbital will have 0 nodes. So I'm going to shade the top lobes of all of these p orbitals. There's no change in sign as we go from one end to the other. I have to draw these a little smaller maybe to fit 4 of them on the board here. So the next one will have 1 node, and we'll put that node here in the middle. So So we're going to change signs as we cross this node. Okay, so that's going to be our next molecular orbital. And then the one just above that, sorry, these are getting a little distorted as I'm trying to fit them in on the board. This one will have 2 nodes. And so we'll put those nodes here and here. So for the shading, I'll draw it up, down, down, and up. Okay, and then the last one, barely be able to fit this in here. This one is going to have 3 nodes. Okay. So here we're going to have up, down, up, and down in terms of our shading pattern. Okay. So our 4 pi electrons are going to go in the 2 lowest energy molecular orbitals. So we'll call this one pi 1. We'll call this one pi 2. This is going to be our HOMO, highest occupied molecular orbital. And then this one is empty, so we'll call that pi star 3. That's going to be our LUMO, our lowest energy unoccupied molecular orbital. And then we have pi star 4. Okay. We have a figure as well we'll take a look at here on the screen that shows the same thing that we drew on the board. 0 nodes, 1 node, 2 nodes, 3 nodes, 4 pi electrons occupying the 2 lowest energy molecular orbitals. These are the bonding molecular orbitals. Anti-bonding, here's our HOMO and our LUMO. They're on the frontier between the occupied and the unoccupied molecular orbitals. Okay, so going back to the board, we told you that electron withdrawing groups attached to the dienophile increase the reaction rate. So that's telling us that in the Diels-Alder reaction, the dienophile is functioning as the electrophile. Okay? So if it's functioning as the electrophile, it is going to be the lowest energy unoccupied molecular orbital that is going to need to overlap with an orbital from the nucleophile. So this orbital right here, the LUMO of our dienophile, is going to be the important molecular orbital to consider on the dienophile when we think about the Diels-Alder reaction. Conversely, over here with our diene, that is the nucleophile in the reaction. And so the highest energy electrons of the nucleophile are going to be the electrons participating or initiating the reaction. So we need to look at the highest energy occupied molecular orbital of our diene. And then we need to ask ourselves, do these orbitals overlap with each other? They have to overlap in phase, meaning the signs of those orbitals have to be equivalent at the places where they overlap. So let's draw this. We'll draw this over here. So we'll draw the HOMO of our diene, 1,3 beta diene. We'll draw that like this. Okay. Then we need to draw the LUMO of our dienophile. We'll put that right here. And it has 1 node. It doesn't matter if we shade up or down as long as we have it opposite and we have that node in place. So I'm going to shade the bottom lobe here and the top lobe here. And what you can see is that as the diene and the dienophile approach each other, the dienophile is going to come either from below or above the diene. We have in-phase overlap of those relevant molecular orbitals. The highest occupied molecular orbital of the 1,3-butadiene will overlap in phase with the lowest energy unoccupied molecular orbital of our dienophile, or our electrophile. Okay? And so because these orbitals overlap in phase, the signs of the orbitals match in the area where they overlap, the reaction is allowed. It will occur. We have a figure from this other textbook that will show that as well. Okay? So here we see the ethylene approaching from below. It can equally approach from above, it doesn't matter, but either above or below. When the carbons of the ethylene approach the terminal carbons of our 1,3-butadiene, you find that those orbitals are in phase with each other. This phrase, symmetry allowed reaction, is not one that we use in our textbook, so we won't dwell on it. But the molecular orbital rules that govern paracyclic reactions, such as the Diels-Alder reaction, are known as orbital symmetry rules. So all this means is that the orbitals are in phase with each other in the regions where they overlap, so the Diels-Alder reaction is permitted to occur according to those molecular orbital theories, those rules, and of course we do see it occur in nature, as we've shown. Okay? So this is the molecular orbital explanation for why the Diels-Alder reaction proceeds, why it works. The missing piece that we haven't taught you yet is how do electron withdrawing groups increase the reaction rate if the electron withdrawing group is attached to the dienophile. So we need to go back and look at the molecular orbitals of our dienophile in order to understand this. So what happens when we attach an electron withdrawing group to our dienophile is we make that dienophile more of an electrophile. Okay? That should make sense. In molecular orbital terminology, when we make it more of an electrophile, what happens is the energy of the LUMO will be lowered. Okay? It's not going to be lower than the HOMO. It's still going to be higher than the HOMO. But the energy of that LUMO is going to drop. It's going to decrease. Okay? And in reactions such as the Diels-Alder reaction, where we have these frontier molecular orbitals on two different reactants overlapping, the gap in energy between those molecular orbitals is related to the reaction rate. The closer in energy those overlapping molecular orbitals are, the faster the reaction will proceed. So if by attaching an electron withdrawing group to our dienophile, we lower the energy of the LUMO, that's going to bring that LUMO closer in energy to the HOMO of the diene. So we're shrinking that energy gap and therefore increasing the reaction rate of the Diels-Alder reaction. Okay? So electron withdrawing groups attached to dienophiles lower the energy of their LUMO, bringing it closer in energy to the HOMO of the diene, and therefore accelerating the Diels-Alder reaction. So you might ask yourself, would the converse be true? Could you add an electron donating group to your diene, raise the energy of its HOMO, and therefore accelerate the reaction? The answer is yes. We don't focus on it in the textbook because, for reasons we won't get into, it's not as common as attaching the electron withdrawing groups to the dienophile, but indeed, if you have electron donating groups on your diene, that will make the diene more nucleophilic and make the Diels-Alder reaction faster. Okay? So that's our molecular orbital explanation of the Diels-Alder reaction. So let's move on to our third point, third rule governing Diels-Alder reactions. And that is the stereochemistry of dienophile, of the dienophile, is retained in the product. Okay? So as you know by now, most of our reactions involve stereochemistry. The Diels-Alder reaction is no exception to that. So we'll take this dicarboxylic acid, diene. The carbonyl groups of our carboxylic acids will function as electron withdrawing groups. You notice that the two carboxylic acid groups are cis to each other on the dienophile. When the Diels-Alder reaction occurs and we generate the cyclohexene product, those two carboxylic acid groups remain cis. Okay. This compound is achiral. It's a meso compound. It has a plane of symmetry. So it does not have an enantiomer, but we will only get the cis isomer, the cis diastereomer of this product, because those carboxylic acid groups are cis to each other. But if we had the trans isomer of our starting material, then we would end up with the trans diastereomer of the product. Okay? And as I draw the trans diastereomer, you'll see that this is chiral. It does not have a plane of symmetry. So it will be produced as a racemic mixture. Neither of our starting reactants is chiral, so whenever we have two achiral reactants forming a chiral product, we always get a racemic mixture. Okay? There are chiral catalysts that can be used in Diels-Alder reactions that can give you a single enantiomer, but we're not going to get into that in this chapter. So, we mentioned this in our last lecture, that the Diels-Alder reaction is stereospecific, meaning that our cis isomer of our dienophile gives us the cis isomer of the cyclohexene product. Trans isomer gives trans product. The reason why the reaction is stereospecific can be understood if we look at the mechanism. It's a concerted mechanism, one step. Both of the new sigma bonds that are being formed are formed simultaneously. And so that does not provide any opportunity for these groups to rotate. And so the relationship that they have in the dienophile has to be maintained in the product because of that lack of opportunity to rotate due to the fact that the two new sigma bonds are formed simultaneously. So that's the explanation for the stereochemistry being retained. Our fourth point involves some terms that we have not learned yet. Our fourth point says when exo and endo products are possible, endo is preferred. So you're probably wondering what the terms exo and endo mean. They refer to bridged bicyclic compounds. Let's take a look at a nice figure from your textbook showing you the difference between a fused bicyclic system and a bridged bicyclic system. In a fused bicyclic system, we have two carbons common to both rings that are connected by a sigma bond. So both the carbons and the sigma bond are shared by the two rings. In contrast, in a bridged bicyclic system, we still have two carbons that are shared by the two rings, but they're not adjacent to each other. They're not connected by a sigma bond. They're connected by a bridge containing one or more carbons. In this case, it's a bridge of one carbon. Here it's pretty easy to see that we have two rings. You might say, how do we know this is two rings? I mean, it almost looks like there's three rings. We have this six-membered ring at the base. You could count a five-membered ring on the left, a five-membered ring on the right. So it's kind of ambiguous how many rings we have here. Well, the way we define the number of rings in a molecule is by how many sigma bonds would we have to break to get back to an acyclic species. And if you look at our bridge bicyclic system, if we broke 2 sigma bonds, we would have an acyclic molecule. So that's why this is referred to as a bicyclic compound. But it is bridged. You see we have this bridge going across. And when we perform a Diels-Alder reaction with a cyclic diene, we end up generating a bridged bicyclic molecule. So let's look at cyclopentadiene. This is our simplest cyclic diene. And we're going to react it with a generic dienophile Z. This is going to be our electron withdrawing group. It could be a ketone or a carboxylic acid or an ester. We're going to heat these reactants up, and we're going to generate a bridged bicyclic compound. These are not the easiest to draw. Okay, but this is what it would look like. So here on the left is the 5-membered ring that was present in the dienophile. We're not breaking that ring up, so it's got to be present in the product. Here at the base, these 6 carbons are the 6 carbons in the newly forming cyclohexene ring. And then this carbon bears our substituent. We can have 2 different diastereomers, where we can have the Z group pointing down, or we can have the Z group pointing up. Okay? And this is where exo and endo, where the terms exo and endo come in. So when you have a bridged bicyclic molecule, if you have your substituent on the side of the larger bridge, so we would say this is a 2-carbon bridge, so we're looking at this carbon, and we'd say this is a 2-carbon bridge and that's a 1-carbon bridge. If we have our substituent closest to the larger of those bridges, that is referred to as the endo product. And where we have our substituent closer to the smaller of those 2 bridges, that is known as the exo product. Okay? So this rule is telling us that the endo is going to be the major product, and the exo is going to be the minor product. Okay? So why is that? Let's take a look again at the screen. There's a really nice figure from the textbook that explains this for us. So we have our cyclopentadiene. We're bringing our dienophile underneath the diene. We could have it above. It doesn't really matter. We've just chosen to bring it in below. And you see that if we have the Z group, the substituent, folded underneath the diene, that's going to lead to the endo product. Okay? Here's our 2 new bonds that are forming in red. Conversely, if we have the Z group pointing away from the diene, that's going to give us the exo product. So if you look at these transition states, you should be able to see that the upper transition state leading to the endo product is more crowded, more sterically hindered than the bottom transition state leading to the exo product. So if you were looking at steric hindrance, you would say this transition state is going to be higher in energy because of that steric strain. So this is going to be the minor product. But obviously that's not the case. So if we're going against what would be observed based on sterics, there must be some kind of electronic reason for that. Okay? And that reason is the Z group is an electron withdrawing group. It's usually a carbonyl group. That carbonyl group has an electronegative oxygen. That electronegative oxygen pulls on electrons, attracts electrons through bonds. It can also attract electrons through space. And so, if you bring that electron withdrawing group close in space to the electron-rich diene, remember we have 4 pi electrons in this diene, you can have an attractive interaction between the pi electrons and the electron withdrawing carbonyl oxygen. Okay? And this is referred to as a secondary orbital interaction. I don't believe this term is used in your textbook, but it is a term that is used to describe these through-space stabilizing interactions. Okay? Secondary orbital interaction between the pi system of our cyclopentadiene, those pi electrons, and our electron withdrawing carbonyl group. I brought some models today, so we can show this to you with our molecular models. Here's the cyclopentadiene. Here's our dienophile. This is our carbon-carbon double bond. This is our carbon-oxygen double bond. The one I made here is a ketone. This is called methyl vinyl ketone. So we're going to bring our diene below our dienophile. This would be the exo transition state because you see that carbonyl group is pointing away from our diene. If we flip the dienophile over, now we have our endo transition state, and you see that it's more crowded. However, we have brought the pi electrons closer in space to our electron withdrawing carbonyl group. So this is our endo transition state, giving us the endo product. And if we were to flip the diene 180 degrees, this is the exo transition state, higher in energy due to electronics, right? It goes contrary to sterics, so this tells us that some kind of electronic effect must be overriding, must be more important than the sterics, okay? So exo and endo apply when we are talking about cyclic dienes giving us bridged bicyclic products. All right, so that's it for our rules governing Diels-Alder reactions. Let's talk a little more about Diels-Alder reactions in synthesis. So I showed you the example last time of how the Diels-Alder reaction could be used to make the pufferfish toxin, the tetrodotoxin. It's important for us to be able to look at a cyclohexene product and dissect it into the diene and dienophile that you would need to prepare that cyclohexene. So if you needed to make a cyclohexene in the lab, it would be important to be able to determine which diene and which dienophile you would need to make that product. So we have two examples of that here on the screen. So what you do is you just you start at the pi electrons of your double bond in your cyclohexene, and you just move the electrons, 3 pairs of electrons around the ring, but recognize that this is in the retrosynthetic direction. And when you do that, you're breaking these 2 sigma bonds, and your diene is going here. So here we would have a 2,3-dimethyl 1,3-butadiene, and we would have a cis dienophile. We have to remember that the substituents on the ring have to have the same relationship with each other. So if we have cis substituents, we have to have cis substituents in our starting material. This is how you would do it if you had a bridged bicyclic product. Here's our 6-membered ring at the base of the molecule that is being formed in the reaction. So we break it open retrosynthetically. We're left with this dienophile, our electron withdrawing ester group. As we'll learn in chapter 18, this chloride is electron withdrawing as well, so we have 2 electron withdrawing groups. And then we have cyclopentadiene as our diene. So it's important to be able to recognize dienes and dienophiles that would be used to make a given product. So we have an iClicker question. Of course, since you're not in class, this is just for practice. So you can pause the video and ask yourself what diene would be used to make this Diels-Alder product. And once you've solved it, unpause the video, and then we'll discuss the answer. Okay, so we start at the pi electrons of the cyclohexene. We move 3 pairs of electrons around the ring, breaking these 2 sigma bonds, giving us a pi bond here and a pi bond there. So we have a 5-carbon dienophile. We have a methyl group on one end. We look at our choices and we see that E is the diene in this particular case. Notice it's drawn in the S-trans conformation instead of the S-cis conformation. Of course, it can rotate about that central carbon-carbon sigma bond and undergo the reaction. If we asked you what is the dienophile that would be used, then A would be the answer, because we would end up with a double bond here, break those bonds. So cyclopentene is our dienophile, and then this methylated butadiene would be the diene. Okay, so it's important to be able to use the Diels-Alder reaction in a retrosynthetic direction as well as the synthetic direction. So we've talked a little bit previously about steroids, very, very important molecules in nature. Steroids have 3 6-membered rings fused to a 5-membered ring. Estrone and cortisone are 2 common examples of steroids. We talked about estrone last semester, the female pregnancy hormone that suppresses ovulation. Chemists have synthesized steroids in the lab, and because we have 3 6-membered rings in steroids, it should not surprise you that the Diels-Alder reaction has been used extensively in steroid synthesis. It can be used to synthesize any of these 3 6-membered rings. Here we have examples where this one is known as the C ring, and that was prepared by a Diels-Alder reaction. Notice that our dienophile is electron deficient because it has these 2 ketones functioning as withdrawing groups. And then that same dienophile used here with an electron-rich diene, 2 electron-donating groups, generating, in this case, what becomes the B ring of cortisone. So Diels-Alder reactions used frequently to synthesize steroids. Okay? All right, our last topic for the chapter is the interaction of polyenes, dienes, and longer conjugated polyenes with light. So we learned in chapters 13 and 14 in our spectroscopy chapters about molecules absorbing electromagnetic energy and being raised from a lower energy state to a higher energy state. So our UV spectrum is fairly high energy radiation. Right? In IR spectroscopy, we dealt with infrared radiation, which is lower in energy, longer wavelengths than visible light. In NMR spectroscopy, we dealt with radio waves, which is even lower energy and longer wavelengths. UV light is shorter in wavelength and higher in energy than visible light. 200 to 400 nanometers are the wavelength ranges for UV light. And it turns out that when you have a conjugated diene, the gap in energy between the HOMO and the LUMO is in the UV range. It's in that 200 to 400 nanometer range. Okay? So the ground state of a diene, we would have our HOMO, our occupied HOMO, and our unoccupied LUMO. If the difference in energy between those orbitals is equivalent to a certain wavelength of UV light, if we shine that wavelength on the molecule, we'll use h nu to represent the UV light, then what will happen is one of these electrons will be promoted to the higher energy level. Okay? So this is our ground state. And then over here, where we have those electrons unpaired, that's going to be our excited state. Okay. And then, of course, the molecule would relax back from the excited state down to the ground state later on. But the absorption of the UV light causes promotion of an electron from the HOMO to the LUMO. And as the number of conjugated double bonds increases, the number of pi molecular orbitals increases, the energy gaps between those orbitals decreases. So the gap between the HOMO and the LUMO will go down, and the wavelength at which those molecules absorb UV light will increase because the energy will decrease. Okay, so let's take a look at that. I said a lot of things there. Let's write it on the board so we can understand. Okay, so here we have 1,3-butadiene, lambda max. The wavelength at which it will absorb UV light and promote that electron is 217 nanometers. Okay, that's at the high energy end of the UV spectrum. Hexatriene, 1,3,5-hexatriene. The lambda max is going to be 268. Okay, about 50 nanometers higher. Smaller energy gap between the HOMO and the LUMO. Okay, 2, 3, 4, 5, 6. Let's see if I've got enough room to draw this one. Barely. Okay, so if we have 6 double bonds conjugated to each other, now we end up 364 nanometers. Okay. More pi molecular orbitals, smaller difference in energy between them, and therefore longer wavelength UV light required to promote an electron from the ground state, or from the HOMO to the LUMO to get from the ground state to the excited state. So let's take a look at a molecule on the screen here, lycopene, 11 conjugated pi bonds. When you get to 11 conjugated pi bonds, now you've shrunk that gap between the HOMO and the LUMO enough that the molecule will absorb visible light, not just UV light. So lycopene's lambda max is 470 nanometers, and that's in the blue-green region of the visible spectrum. So lycopene absorbs blue and green light, and it reflects red light, and so substances that have lycopene in it will appear red. And what has lycopene? Tomato juice. Tomatoes have lycopene. Lycopene is what makes tomatoes and tomato juice red. There's a great demo that I would do for you if you were here in class, but since you're not, and since the demo lab is closed today, and I can't do it for you on film, I will show you a link to the demo that someone has done online. If you add bromine to tomato juice, you will brominate the alkenes and you'll disrupt the conjugation. And when you disrupt the conjugation, you're now going to increase the gap between the HOMO and the LUMO, and you're going to change the absorption. It's not going to absorb the visible light anymore, so you're going to remove the color. The colors are going to change. The demo is called the tomato juice rainbow, because as you add the bromine to the tomato juice, as that bromine sinks down through the tomato juice and reacts with those double bonds, it decolorizes it. And it does so in a way that it goes through a different spectrum of the colors. So it's a really cool demo. I encourage you to click on the link that I will put on Learning Suite and take a look at the demo. Okay? One other feature of this is that as we promote an electron from the ground state to the excited state, we're taking an electron from a bonding molecular orbital and putting it into an anti-bonding molecular orbital. So we're actually going to break the pi bonds and allow rotation about the carbons that are involved in the pi bond in the ground state. In the excited state, we don't have that pi bond, so we can rotate. And then when it relaxes back down to the ground state, it reforms. And that is what forms the basis of fission. So let's take a look at this figure here that shows us the molecular basis for fission. We've told you before that carrots really do help your vision, and this is why. Beta-carotene is an incredibly conjugated molecule. It's responsible for the orange color of carrots. When you ingest beta-carotene, enzymes in your body will chop this molecule in half, oxidize it, and turn it into trans-retinal. Then enzymes in your eye will conjugate this aldehyde of retinal with a nitrogen. This is going to be on a side chain of a protein called opsin. and generate a protein small molecule conjugate known as rhodopsin. You'll also notice that this enzyme is able to isomerize chemically one of these double bonds from trans to cis. Okay? We can chemically isomerize double bonds, and the enzyme is able to do that. This is a highly conjugated molecule. It will absorb visible light. When visible light enters your eye, what it does is promote an electron from a bonding orbital from the HOMO to the LUMO. That breaks the pi bonds, allows rotation at that excited state. This is sterically hindered. We've got a kink in the chain. So when it's allowed to rotate, it's going to rotate to unkink the chain. And then when the molecule relaxes back down to the ground state, we now have a trans double bond. That's a pretty massive change in conformation when we go from this cis double bond in rhodopsin to the trans double bond. That change in conformation triggers a nerve impulse that is then recorded in the brain and turned into vision. I don't know how that part of the vision process works, but I do know how the molecular basis works, how the light is then converted into a nerve impulse, because that light promotes an electron from the HOMO to the LUMO, allows rotation about the carbons that were involved in the pi bond in the ground state, and then when it relaxes back from excited state to ground state, it's now trans, conformation is changed, nerve impulse, vision. Okay, so eat your carrots so that you have plenty of beta carotene, plenty of transretinal available in your eyes for this process to occur. All right, so that's it for chapter 16. In the last few minutes of today's lecture, let's introduce benzene. Okay? Benzene is a very interesting molecule. Okay? It was originally discovered in coal tar in the 1820s by Michael Faraday. If you've heard of Michael Faraday, you probably associate him with electricity or physics. Back in the 1820s, there were plenty of scientists who did both physics and chemistry, and Michael Faraday was one of them. So he discovered benzene in coal tar. Other chemists found a molecule that they called benzoic acid in plants from extract from trees, and they converted benzoic acid into benzene. That's where its name comes from. Benzoic acid is related to the name of the tree where the molecule was first isolated, and then benzene was derived from benzoic acid. So in the 1860s, we had chemists who were doing experiments to try to figure out the structure of benzene. They knew the molecular formula. They knew that the molecular formula of benzene was C6H6. At that point, they knew that carbon formed 4 bonds, and so they knew that there were 4 degrees of unsaturation in benzene. There's no way you can have 4 rings with only 6 carbons. That's physically impossible. So they knew there had to be a whole bunch of double bonds in this molecule, and they knew that molecules with double bonds underwent addition reactions. So somebody, I'm not sure who, but some organic chemist tried to react benzene with bromine. We know, I showed you the demo in class, we know that alkenes react instantaneously with bromine. So they were shocked to find that there was no reaction when they reacted benzene with bromine. Somebody then decided to add a catalyst. They added iron tribromide. This is a Lewis acid. It catalyzes the reaction of bromine with benzene. So they got a reaction to occur in this case, but it was not an addition reaction. It was a substitution reaction. Okay? They replaced one and only one of the hydrogens of benzene with a bromine. Okay? So that was shocking to them, that they saw substitution and not addition. Another interesting feature was that they only obtained one product. There were 6 hydrogens on benzene, so you could substitute any one of those 6 hydrogens with a bromine, and they only got one product. So that told them that all 6 of the hydrogens of benzene were equivalent to each other. Okay? So they could perform other substitutions on benzene, but no additions. They couldn't perform any additions. So there was something about this unsaturated structure of benzene that was stable and unreactive. And this was intriguing chemists back in the 1860s. So in 1866, there was a chemist named August Kekulé. He worked in Berlin. Okay? So August Kekulé was trying to figure out the structure of benzene. And he started with what was known. It was known that the molecular formula was C6H6. It was also known that all hydrogens in benzene are equivalent. I just explained to you how that was known. And it was also known in the 1860s that each carbon has 4 bonds. Okay? So he took these facts and he tried to figure out the structure of benzene. Now here's where the story gets interesting. It's unclear if Kekulé is making this up to make the story interesting or if it actually occurred. But it's a cool story, so I'll tell you how the story unfolds. So Kekulé says that he was thinking about this in his study at home, and he had a fire going. It was cold, probably in the wintertime in Berlin. And he fell asleep. And while he fell asleep sitting there, he had a dream of whirling snakes, intertwined snakes, that were biting their tails. Okay? Now, before you think he was high on something, this idea of intertwined snakes was a symbol of alchemy, and so it was something that was prominent back in the 1860s. So it wouldn't be a big stretch to dream about these intertwining snakes. But when Kekulé woke up, this idea of these intertwining snakes was in his head, the snake forming a ring by biting its tail. And that gave him the idea, or so he says, that benzene was a ring. In the 1800s, there was no concept of cyclic molecules. Right? They thought that they hadn't considered the fact that you could have rings. They thought that the atoms were just connected together in chains. So Kekulé was the first person to come up with the idea that a molecule could be a ring. And so the structure he came up with for benzene looked like this. Okay? Now Kekulé was an architectural student, and so he probably looked at shapes a little differently than most chemists did. So that architectural background probably helped him come up with this structure. Resonance theory was not understood at the time of Kekulé, so he thought that benzene was 2 rapidly interconverting isomers with alternating single and double bonds. Okay? That was his idea. So he was correct with one exception. Later on, it was when the ability to measure bond lengths came about, it was determined that the carbon-carbon bond lengths in benzene are identical. 1.40 angstroms. So all of the carbon-carbon bond lengths in benzene are identical. So obviously you could not have 2 interconverting isomers that would have 2 different bond lengths. And so now, because we have resonance theory, we recognize that these are resonance structures of benzene. Okay? We can draw our curved arrows connecting the resonance structures. 2 equivalent resonance structures gives us a hybrid. There are 2 different ways these hybrids are drawn. Sometimes they're drawn with dashed circle in the middle. Sometimes they're drawn with a full circle. So these are hybrid structures of benzene. But most of the time we'll actually see the resonance structures because it'll be easier to understand how reactions occur with benzene. But we know that the bond lengths, carbon-carbon bond lengths in benzene are identical. Incidentally, this is exactly halfway between the standard bond lengths for a carbon-carbon sigma bond and a carbon-carbon pi bond. So it is a 1.5 bond as we would expect. We have a figure. Let's go ahead and go to chapter 17 PowerPoint here. And benzene is conjugated. Each of the 6 carbons in benzene has a p orbital. So it's fully conjugated. The 6 pi electrons are delocalized across all 6 carbons of the ring. So we see that here. We see this electron-rich region above and below. We can't see it below, but we see it above in this orientation. due to those pi electrons. The last thing I'll show you is a model. Here's a model of benzene that I made for you. We see our 6 p orbitals. I put rubber bands on them to show the individual pi bonds. If you were looking at one resonance structure, the reality is instead of the individual pi bonds, we have those 6 pi electrons delocalized and spread out across the entire molecule. But all of the bonds, the carbon-carbon bonds in benzene, are equivalent. Same bond length, same bond order. They're 1.5 bonds. Okay, so that concludes our recorded lecture for today. And I look forward to seeing you in class on Monday, ready to take the quiz at the beginning, and then we'll continue on with our discussion of benzene afterwards. Thank you.